but I will talk a little bit about the Rush stuff that Patricia Boyle and Bob Wilson have done with respect to the reserve from before. But I'm also going to show some new data that we are working on. It's still in the raw phase. It's not published yet, but it was done as part of an NIH grant, and we thought it was kind of interesting what we found. So it might be good to share before it gets published. So this is sort of a mix of little Rush-Ross map interaction with CHAP. I don't know if you guys know CHAP, the Chicago Health and Aging Project. So I essentially work with Dennis Evans at CHAP, and we also worked with Lisa Barnes and Bob Wilson and David Bennett and everyone else. So I want to start off my talk by acknowledging all the great mentors that I've had in the years past. Dennis Evans, David Bennett, Bob Wilson, Lisa Barnes, a neuropsychologist, Patricia Boyle, and Charlie, who's helping us with our MRI data for the CHAP study. We have about 1,000 MRIs that we did in CHAP as well. So we have a lot of different moving parts, and we want to sort of connect these parts. So you guys have already seen a lot of these background slides, which kind of talks about what is this concept of reserve/resilience. Typically, it's being defined as preserved cognition in the face of challenges, the challenges being defined in terms of autopsy studies and pathology, or pathology from neuroimaging studies. But we started to ask the question, what if we sort of don't have the pathology studies, and what if we don't have neuroimaging studies? What can we do to get a definition or a working definition of resilience? So we went about doing this slightly differently and using completely epidemiologic data without any neuroimaging and neuropath. But the beauty of this is we also have neuroimaging and neuropath data. So it will give us a way to sort of validate some of these measures and cross-check and cross-compare how these measures do in different populations. And our population is mostly has a very big African-American concentration. It's about 65%. So it's a highly African-American population. And our cognitive battery was done in almost 10,000 people. So we have these huge amounts of cognitive neuropsych data, and we wanted to use that, not just use a smaller subset in terms of neuropathology or neuroimaging. So we came up with this definition that we're going to look at cognitive changes and define resilience based on these cognitive changes as to people who show least cognitive change or slower cognitive change in the face of these challenges that they have. We defined 4 general categories of challenges: social, economic, psychological, and physical factors. Essentially, these are known risk factors for AD and for cognitive decline in general. And they can be derived in population studies, essentially collected in all population studies that don't have pathology, or using either autopsy or neuroimaging. And we can, given that our study has both neuroimaging, pathology, and the cognitive measures from epidemiologic data, we would be able to compare these 3 different approaches and sort of get a sense of what might be happening, and if we can use these measures in larger population studies that don't have these measures. And finally, one of the goals was to actually identify lifestyle factors and other risk factors like cardiovascular disease and genetic risk factors that might be related to this concept of cognitive resilience or cognitive changes over time in the face of challenges. So as everybody has probably seen this 1,000 times and understand this concept really well, cognitive changes happen over a lifespan. And typically, cognitive changes occur in small increments. They go down slightly over the years. There are a subgroup of people who show faster decline and go on to become MCI. And once they hit the dementia threshold, which is usually a binary classification, they are deemed as AD or something related to dementia, to vascular dementia, based on what sort of pathology they observe. So essentially, cognitively normal people, people who show some moderate decline, who are classified as MCI, and people who show drastic changes in their cognition. So there's a lot of research that's been done in terms of what are the factors that are related to these different processes, in terms of the decline, in terms of the levels, in terms of the age at onset and the onset of the change itself. So typically, I'm pretty sure all of you know this already, but it might be useful just to sort of touch on what our ROSMAP colleagues have done in the past, essentially define AD as progressive loss of memory functioning and other cognitive abilities and functional impairment. So essentially, the concept is about the progressive loss. It's about the change in cognitive function, like how do we capture this change which makes sense in terms of the pathology itself. And the AD pathology is defined by the presence of plaques and tangles in the brain. And when the pathology disrupts the ability of the neurons to communicate with each other and function properly, it leads to more disjoint parts of the brain and leads to higher AD pathology. So that's the basic thinking. So David published this study in 2005. I'm pretty sure everybody has seen this. There's 99% of people in ROSMAP had some form of plaques or tangles. It had varying degrees, so essentially everybody that they saw in their clinic postmortem had some form of plaques or tangles, and 36% had at least one or more microscopic infarcts, and 28% had one or more microinfarcts, and 10% had neocortical Lewy body. So essentially, if you can see this is more of a mixed pathology sort of definition of what happens in the brain. So essentially, everybody shows some pathology, but it's not clear how that pathology actually leads to AD. And David also reported the concordance between clinical and pathological diagnosis, which is very interesting. There's almost 90% concordance between the two, which suggests that in terms of the clinical definition, you're capturing a lot of these changes in pathology, even though it's not clearly stated. Okay, so next we just sort of want to understand what is the relationship of the pathology to decline itself. I'm pretty sure a lot of you know this already, but essentially 41% of cognitive decline is explained by pathology, and I think Dan talked about this in the morning too. The contributions of amyloid, tangles, and gross infarcts pretty much accounts for most of this variation in cognitive decline. And interestingly, only 25% of the variation is explained about the onset of the cognitive decline itself. So less is known about the onset and more is known about the variation in the disease itself. So this was published by Patricia Boyle in 2013. Okay, so where do we go from here is the question. So we saw that if you had the pathology data, you can essentially understand cognitive decline in terms of what's going on in your pathology. But the problem was pathology is measured only after death. So essentially you have no crosstalk between cognitive decline and pathology. Neuroimaging can be used to define this crosstalk, which I think Dan and Bruce Reed pretty much did, where they looked at changes in cognitive functioning and how it related to the changes in neuropathology. But our question was, okay, what if we came up with a different solution where we pretty much explain the same amount of variation, but maybe not the same variation, but maybe a different aspect of the variation that can be captured by this scoring algorithm? And would it be good enough? Like, how would it relate to what we already know in terms of neuropathology, in terms of resilience measured by neuropathology in autopsy and neuroimaging studies? So we went about just trying to experiment to see if we can sort of understand this concept. So what we did was we had two main objectives here. First objective was essentially to understand cognitive changes and see how people showed slower cognitive changes despite having these high epidemiologic risk factors, essentially defined through social, economic, psychological, and physical factors. And then we wanted to see, since we had this huge population of African Americans in our sample, do these vary by race? Because that's one of the primary questions we want to ask. Because we know that African Americans have higher adversity in childhood, and they have different exposure to life course measures. So it's really important for us to at least understand where these differences, if any, might be impacting the resilience, and if so, does it impact both in blacks and whites equally? So I don't know how many of you know the CHAP study design, but it's very similar to YCAP and ROSMAP. Essentially, ROSMAP essentially collected about the same amount of data in about 1,500 people. I want to say 1,800 people annually over a span of 20 years, and they're still following them. CHAP collected the same data essentially in 3-year cycles. So the study started in 1993, ended in 2012. We have 5 follow-ups, 6 cycles of data collection. But the good part is we have a huge sample. We have almost 10,800 people that came for baseline assessment. We have 7,500 people with 1 or more cognitive assessments, and 4,900, 5,000 people essentially with 2 or more cognitive assessments. And the best part is we actually have neuroimaging done on almost 1,000 people, 300 repeats, and I want to say 150 that had 2 or more MRI measurements. So we have a whole set of data set that we can essentially look at these different concepts and try to parse out what might be happening and what might not be happening. So briefly, talking about the measures that we used, we had social and economic measures, social measures starting from childhood onto midlife, onto late life. We had a composite measure that collected both childhood SES, midlife SES, adult SES, and of course years of education, which essentially happened in early life. And we also had measurements on social engagement, social measures like social engagement, social networks, social diversity. We have shown in the past that these measures are related to cognitive decline, and we were not sure if these measures protected against these cognitive declines in any way or form. We also collected psychological measures like depressive symptoms, perceived stress, and perceived discrimination, which is more relevant for the African American population. Essentially, Lisa Barnes showed differences in how these measures might be related to cognitive decline. And in terms of physical functioning, we essentially know that there is a very high association between cognitive changes and physical changes. Essentially, one predicts the other in almost a cyclic pattern. So we have longitudinal data on physical functioning scores that use a 3-measurement scale in terms of a tandem walk, chair stand, and timed walk. And we also have a boatload of functional limitations on ADLs, IADLs, the NAGI, and the Rosso-Breslow scores and whatever not. But for this, we just essentially used the ADLs only because it was fairly a robust measurement. And we have shown that ADLs are related to cognitive decline in a more strongly fashion than other measures. And of course, all the age, race, demographic characteristics were measured using the 1990s U.S. Census-based questions. Since the study started in 1993, everything was sort of well-defined and consistent through the years. So, in terms of neuropsych tests, there were 2 different sets of tests that I did not talk about. One set is called the population interviews, where a short battery was administered during in-home assessments, and there's a longer battery that was given as part of the clinical assessment. We're going to focus on the short battery test because we've generally shown the short battery and the bigger cognitive battery actually correlate fairly well, and they predict AD at about the same level. So the short battery is based on 3 pieces. How am I doing in time? Okay. The short battery is based on 3 pieces: the perceptual speed scores that we're using simple digit modalities test, the East Boston memory test for immediate and delayed recall, and the MMSC. So Bob came up with this really nice concept that you might be able to combine these scores and come up with a composite score, and the composite score explained for about 76% of the variation in each of the individual tests. So the tests were standardized, and they were referenced to the baseline population, essentially giving some sort of interpretation in terms of change in standard deviation units, which is sort of very standard as what's being done right now. Okay, a little bit on the demographic characteristics of the population. There are definitely some racial differences in both the epidemiologic factors and in terms of cognitive changes and cognitive function itself. So we wanted to first understand what these factors are and how they might be changing over time as well. So in terms of age, you can, of course, see that African Americans are almost 3 years younger than than European Americans, and they had less education by about 2.5 years, and their cognitive function was definitely lower as well by almost like 0.3 standard deviation units. But there was no differences in terms of gender. Sex differences were sort of non-existent. Essentially, it was 60% females in both African Americans and European Americans. Okay, so we sort of know what the population looked like. So we wanted to next go ahead and see if we can create a score for cognitive resilience based on this population using epidemiologic data. So we used a residual approach, which was shown earlier. So essentially, a lot of people have talked about it. We didn't use the interaction or the moderation approach. So essentially, we used multi-level, multivariate linear mixed effects models with subject-specific intercept and slope, essentially capturing the variability in subject level of cognitive functioning and their changes over time. And we essentially accounted for or adjusted for it more appropriately for social, economic, psychological, and risk factors in the model. So essentially, we put all the factors that we know were related to cognitive decline, and we put them all in this multivariate, multi-level linear regression models. And we essentially got subject-level, time-specific residuals. So essentially, at each time, you get a residual for each person, and then we sort of aggregated those residuals. The residuals typically vary from -3 to +3, since the scale of the cognitive function was -3 to +3 in general population. And using those residuals, we marginalized the scores to a normal distribution and created a score that ranged between 0 and 100. And then we looked at the distribution of the scores, and we essentially split the scores into 4 groups just to describe what the population looked like in these different groups. So we classified them as low, medium, moderate, and high resilience groups, and interestingly, we were able to explain about 40% variation in cognitive decline, much similar to the autopsy and the neuropath studies. Yes? It's a longitudinal change in longitudinal cognitive function, yes. Decline in cognitive function. Okay, so then finally we'll want to see how the resilience scores differed between blacks and whites. I'll show you the distributions and whatever not. So after aggregating each individual's residuals, we were able to come up with I don't know if it's nice looking, but still it seems like a little bit long tail on the left side, normal distribution. You can see it peaks about between 0 to 1, and you can see the distribution is at least like, it's not wacky for each individual. Yes?
Can I ask probably a very silly clarifying question? How do you standardize a subject-specific residual?
So we took the residuals, we averaged them, and we got an average residual per person, and then used the per person residual to use a normal transformation using the mean and variance of the reference distribution, and we flipped it around. So it's essentially a p-norm of the reference distribution. So this is what the residual distribution looked like, and I will show you what the normalized distribution looked like. So essentially, it's capped on the both ends due to the censoring on the normal distribution, And you can see this little bump. These are essentially people who had, according to our estimate, a low cognitive resilience or higher degree of cognitive change, even though they had about the same epidemiologically defined risk factors as the higher group. I'll show some data on how the groups compare next. But essentially, we were able to come up with a distribution of what we defined as epidemiologically defined resilience. And you can see that the blue defines people who had better cognitive function, even though they had similar risk factors, similar to the lower group, and the yellow and the orange correspond to people who showed moderate decline, but not as high risk factors. Okay, so moving on to the next slide. So essentially, the four groups that are defined here, this is essentially a continuous scale. We said, okay, we're going to break the scale down, get some estimates of what it looks like in the population for the four groups so we can do some comparisons. So the blue line shows the people who had, according to our estimate, the best resilience measure. So they started high and they declined, but they did not decline that fast. Essentially, I'll show you in the next slide what the decline actually was. And green slide shows the next set of people who showed moderately lower levels and showed about slightly higher decline than the high cognitive resilience group. And the orange and the red corresponds to the people with the low decline. They had the lowest scores, and they declined the fastest. Okay, so let's look a little bit about the group characteristics, see how the group characteristics look. Is it age, or is it something else that's driving these differences in levels and decline, or do we actually capture some aspects of these characteristics fairly well? So if you look at I'm just going to give you a short comparison between the low and the high groups. The low and the high groups have about the same approximate age. They're both about 72 years old. So it's not, definitely the decline is not due to some age effect. Education, definitely yes. As we have seen in the past so many times, education is related to levels. So essentially the difference in the levels is described by almost 3 years of education. So you can see that the low group had about 10.4 years of education compared to the high group that had about 13.6. And it's almost graded where the resilience measure seems to show some improvement with increases in education. And our global cognitive function essentially was lower due to the lower education. It was negative 0.48, and it was higher in the high group, 0.86. And if you look at the change, or the delta CF, which is the change in the cognitive function, I call it the GC change, the global cognitive change, you can see that the low group showed the highest cognitive decline, 0.081 standard deviation units, almost 3 times higher. That of the high group, which showed 0.026. And you can essentially see that the medium and the moderate groups had very similar, but they were almost graded in terms of low, medium, and high. Again, there are differences in memory scores and speed scores and in MMSC that as one would expect, because the global cognitive function is definitely different between the groups. And you can also see that the depression scores are pretty much balanced. It's about 1.69 in the low group, and it's about 1.57 in the high group. They're pretty close to 1.6 and 1.7. Physical functioning is pretty much dot on. It's 10.3 in the low group and 10.2 in the high group. So they are functioning. They have similar depression. They have similar ADLs and IADLs. You can see it's 0.84 in the low group, and it's 0.80 in the high group. They're essentially very similar in terms of their physical characteristics. Yes?
Do you have E4?
Do I have what?
ApoE.
Yes, we have ApoE4.
Do you know how it breaks out?
We did not look at ApoE4 yet. Females, you can see almost show a nice graded relationship, so all you women here are in better shape. Low resilience, about 50%, high resilience, about 75%. It's almost graded. You can see the nice little jumps happening, 54, 57, 66 to 75. And interestingly, African Americans, the group that we are really interested in, also show very similar prevalence between low and high resilience groups. About 75% of African Americans are in the low group, and about 76% are in the high group as well. So there's something interesting going on in terms of race. What might be the reasons for these highly resilient people? Are they factors that we can identify, which can lead to better interventions and whatever not? So there's definitely some race-specific differences in terms of the medium and moderate. We don't see that high of a difference, but again, we traditionally know that African Americans show lower levels and slower decline than European Americans. Yes?
Just as a reminder, the global cognition, the measures that go into it, are they largely verbal, or are they a mixture of verbal and
So they're a mixture of the speed scores, the executive functioning, episodic memory, and the MMSE. So they're essentially a combination of the three. Okay. So in terms of resilience in African Americans, the distribution looks pretty much like the original piece, where you can see this little blob on the left side. They're essentially African Americans with lower levels of cognitive function. We know that African Americans traditionally test lower on the neuropsych test, so it might be picking up these differences in the neuropsych test to begin with. But what's impressive is this higher group, which is the blue group, is bigger in African Americans too. So let me just go to the next slide. You can see the difference between the European Americans and the African Americans. They definitely have a shorter tail on the European American side, but they have the bigger bars on both the medium and the moderate. But if you look at African Americans, they definitely have a higher resilience in that group as well. So we want to essentially at least understand what are the factors that might be related to this high resilience, and what are the factors that might be related to this low resilience in African Americans, and how they might be different for the two races, if they are different, or how they operate differently. So I don't know how I'm doing in terms of time. 35. I think I'm sort of almost there.
12 minutes.
Okay. So what we did essentially was we derived a summary measure for defining a cognitive resilience scores using epidemiologic factors, essentially rating from a 0 to 100. The basic idea is that better cognitive function and slower cognitive decline can be seen in those who are more cognitively resilient than those who show similar epidemiologic risk factors. And the good thing about this is we can essentially derive this in other epidemiologic studies. So the way that we envision this is we're going to add new pieces to this data. This data is fairly new. We haven't published. We haven't even shown this data outside the group. I think other than Jennifer Weave, nobody else has seen this data. So it's still in the infancy stages. We haven't done a lot of testing. We haven't done a lot of more prep work and understanding how these different factors might be related. But we might be on to something interesting that could be potentially used to define. But again, we have to we have a lot more work to do in terms of understanding what's coming out of the score. And we also have neuroimaging data, which we just received from UC Davis with the Charlie's group. And we have done some preliminary work on looking at how resilience that's being defined using the neuropath, white matter, gray matter, and other neuroimaging components, how a similar score would correspond to this epidemiologic measure. We have some data that shows that the concordance is about 75 to 80%, depending on which factors you see and how you define these factors. We're still in the process of coming up with a little bit more, I want to say, flexible way of approaching the neuroimaging data, because we believe there might be variations in African Americans and European Americans that might be different, and how we might be able to capture that. So we have some preliminary data which suggests that the concordance is fairly high. Next up, we are actually doing a very similar analyses in ROS-MAP, because ROS-MAP has autopsy data. They have all the epidemiologic data similar to CHAP. So we are in the process of looking at the concordance between autopsy-driven resilience measures and epidemiologically driven resilience measures using ROS-MAP data. So that'll be the next step. And the eventual goal is essentially to understand how these different lifestyle, cardiovascular, genetic, ApoE risk factors are different in these groups of resilience and how we might be able to use them going forward. And our primary goal, as always, has been to understand racial differences, and our cohort is essentially powered to understand a lot of these racial differences in terms of different measures, cognitive decline being one, and cognitive resilience now being the other. So that's all I have right now, but I'll take a lot of questions, I suppose.
All right.
Thank you. Oh, thank you, guys. Okay.
Hi.
I'm going to get creamed. I know, Jennifer, I'm going to get creamed, so go ahead.
So that was very thought-provoking. I am confused, but I think it's just because I'm trying to catch up with what you did. So maybe through this question, it'll help me narrow down.
Sure.
Did you have some hypotheses? Did you have some hypotheses about how cognitive reserve, or what I think you sort of use the terms interchangeably,
Yeah.
resilience and reserve. So did you have hypotheses about how resilience or reserve might be expressed or might act differently across race?
Yes. So, yes, we did. So we have lots of data that was published in the last 10, 15 years, which suggests that some of the factors that are observed, for example, education. Lisa has a very neat paper which shows how education impacts cognitive decline differently in blacks and whites. And we have another paper that Lisa's published in neurology that also looks at how early life adversity in terms of amount of food, nutrition, that relates to cognitive decline differently in blacks and whites. So going in, we sort of had this hypothesis that these factors are going to behave differently in blacks and whites. We know that some of the factors that we already looked at are like education and these early childhood measures. Did I answer your question? So yes,
No.
going in, we sort of know that.
Okay, but I'm giving you more questions as I stand up here. I don't want to dominate. I'm just going to ask one more thing.
Sure.
Which is that we know that African Americans are more likely to have these risk factors that you put in your model to define your residual.
Yes.
Okay, they have lower SES, they have lower education, they're lower income, less high occupation.
Yeah.
That's a main effect to me. If you
Okay.
look at, that's a main effect. Are you suggesting that there's something that is an interaction that
With time, yes. So is that okay? Go ahead. Finish your question.
All right. It's never good to interrupt me.
I'm sorry.
Because I don't remember what it was I was going to say. It's not you. It's just so.
Short-term memory.
Yeah. So the interaction would be that low occupation is bad for everybody, but even more so among African Americans. Or maybe it's the interaction is such that low occupation is even more harmful in terms of cognitive decline for non-Hispanic white people. That because so I'm just trying to get a sense of of that.
Okay.
Saying it's a main effect or whether it's an interaction.
So that's a great question. As usual, we do our approaches two different ways. We used a more combined sample, and then we stratify the sample by race. Essentially, when you stratify the sample by race, you're accounting for a lot of these interactions out. And we have seen that the interactions are quite different. They operate differently in blacks and whites. For example, the SES, midlife, occupation, personality measures, they do operate differently in blacks and whites. So we have been able to at least account for some of these variations by using interactions in our approach. Maybe it's not the best approach at this point. Maybe we should look at these measures stratified by race and try to see where these differences might be happening more in blacks and whites. That's a good question, and I think we're going to look at it next. But we have looked at it stratified by these risk factors and changes in cognition before, and we have found differences.
Hi,
Yeah.
Annie Racine. I want to use your talk also as an excuse to get clarification on some of the concepts.
Okay.
So we're talking, and Jen just sort of alluded to this, that you talked about cognitive and resilience and reserve kind of simultaneously. And so I think what I understand from Laura's talk is that when we're talking about cognitive reserve, we're talking about cognitive performance given a certain level of brain integrity or brain pathology. Whereas cognitive resilience is more talking about cognition, cognitive performance at a certain level of, in the face of adversity and risk factors.
Yes.
So I think what you're talking about is cognitive resilience. So first off, actually just like Laura, is that right, what I just said? Yeah, okay. So,
You have, sorry, can't you have resilience to neuropathology? Is that really how we're going to split it up?
And
What?
I thought resilience was that resistance was you don't get the brain pathology. Right? So that's a brain.
So okay. Next question. Let's move on. Okay.
So what's the answer, Rob?
I don't think I think you guys are more of an expert in this than I am. I am very new to the resilience/reserve. At least the way that I see the distinction is in terms of the approach. When I think of reserve, I think of what's left in your tank. When I think of resilience, I think of how far can you go with what's left in your tank. So they are two different questions, but they sort of capture different aspects. So I don't know what the answer. I don't think there's a correct answer here. But yeah, it's. I'll let you guys decide what you want to call it.
Yeah, so
Last word, Paul.
I'm Paul Crane from the University of Washington. It's a nice talk, and these are complicated issues. But I do think that the language we use is important. So if we're defining, as Laura did, resilience on the basis of you have brain pathology and you do better than expected, you don't have that. We have demographic predictors and risk factors and stuff, and people doing better than predicted on the basis of risk factors. But you can't then say which parts of that are because they don't have the brain findings, they don't have the pathology.
Okay.
So I think it would be helpful to use different language. You absolutely have a predicted cognitive performance on the basis of a bunch of factors and a residual from that. But that is not the same as having those predictors be brain pathology related predictors. Does that make sense?
That makes sense, but again, you need to remember what exactly is the purpose of doing this. Do we have brain pathology on everybody you see in your study, or those that are deceased? Functionally talking, you're looking at a disease status after somebody has died. How much can that be used to define somebody who's living? I think that's a different question. But you can't get the data you want until you're essentially dead, which begs the point, what can you do with those people who are alive? So we're not saying that this is going to replace the pathology framework. We're trying to say that there is an alternative that we should look at. It's not, you can't really substitute pathology with risk factors, absolutely. But these risk factors have been studied in pathology studies too, and they correlate fairly well. So there's some precedence for it, but it's not defined using the pathologies already there in people who are diseased. So, or using neuroimaging studies, which are still kind of not clear in terms of the variation. How much do they pick up? What parts of these disease are we actually measuring and whatever not? So, yeah. I do acknowledge there are limitations to it, but this is the best we could do with what we have.
Thank you.
Thank you.